name is naked guy. Make sure you know naked guy, okay? And apply directional terms to the naked guy. Make sure you know him. Okay, now, let's see if I made any mistakes here. So here we have 5 kind of blank things. You're not gonna write down anything. You're going to pick A, B, C, D, or E. It's a perfect matching situation, right? What I would do, I would just draw a cross, whatever you wanna do, mark up the exam. By the way, I never give you any pictures. No pictures, no diagrams, that's all written, okay? No pictures. Now, so let's do this. The femoral region. Where is the femoral region? Bam, right? Femur, femoral region. Where are your tarsals? Ankles. Okay, so the femoral region is what to the tarsal region? Proximal. Can you say superior? Yes, but you would get it wrong. Okay, no. Remember, upper and lower limbs, we're using proximal and distal. All right, yes, awesome. The mental region, where is the mental region? Your chin. So the mental region is what to the umbilicus or the belly button? Beautiful. Can you say cranial? Yes. Can you say cephalic? Yes. If you switch it up, you could say that the umbilicus is what to the mental region? Caudal, beautiful. Caudal or inferior. Know all the options. Now, patellar. Ah, you know where patella is, right? That's your kneecap. Patella's up here. And popliteal region or popliteus behind the knee. Patellar in front, popliteal region in the back. So we're talking about lower limbs. Should we use proximal and distal? No, because these two things are the same distance from where the whole thing attaches. So, what's my question? The patellar region is what to the popliteal? Ventral, can you say anterior? Yes. Switch it up, popliteal region is what to the patellar? 2 words, please. Posterior dorsal, anterior ventral. Yeah? Okay, make sure you write all that stuff down when you study. I'm a writer, I write everything down. Antebrachium is what to the brachium. So where is your antebrachium? You're going like this, bam. Antebrachium, where's your brachium? Brazo. So what's the order? Beautiful. The antebrachium is distal to the brachium. Let's do another one. How about the metacarpals are what to the carpals? I'm not going to be showing. So, yes. Okay. Now, the pollex is what to the 5th digit? I'm lying. Don't be trusting me. Lateral. Because in this position, am I an anatomical? No. Switch it up. Write it down. The pollex is lateral to the 5th digit. I'm not going to take my shoes off, but the hallux is medial. The hallux, big toe, is medial to its 5th digit. Pollex, lateral. Probably won't do specific bones, but here we go. Here's the radius. Here's the ulna. Fill in the blank, please. The radius is what to the ulna? Lateral. Because you're in anatomical position, right? Okay. Okay, the liver and spleen. Liver and spleen, are they on the same side or opposite? Opposite. Yeah, so they're contralateral. Yeah. If I said the liver and the gallbladder, they're what? Ipsy. How cute is that? Ipsy, same side. Contra, different. Here's another one. Ascending and descending colons are what? Contralateral, right? Pretty much give you all the questions over here. But make sure that, here's another one. The olecranon region is blank to the antecubital region. Naked guy, he's very cute. Where's your olecranon region? Bam, right here. Olecranon, this is the olecranon process. Antecubital, I didn't say antebrachial, antecubital, in front, right? So the antecubital to the olecranon, antecubital is, or the olecranon is a dorsal, beautiful. Anterior ventral, posterior dorsal, kind of elongate the dorsal, AB, anterior ventral. Ventral means belly. We're good with this? Yes, okay. Now, the bottom one could not be easier. Okay, so as far as this chapter 2, right, do we even have to go over this? Probably not. Mitochondria, the heading will be, please match the organelles to the correct description or function, right? Okay. Mitochondria, what do? What's the best name? Yeah, so no mitochondria would be ATP. So number 6, mitochondria would be energy, right? All right, so that would be synthesize ATP or makes energy, something like that, powerhouse. Okay, so mitochondria is E. I would do, on my paper, I'll just match it all up. If you want to do that, that's fine. SER, smooth endoplasmic reticulum. What's the best? Calcium storage, especially in muscle cells. What if I had, instead of calcium storage, lipid metabolism? Same thing, right? Okay. Rough ER, what does it do? Makes proteins, absolutely. Lysosomes, yeah, absolutely. Acid hydrolases, anything that ends in an ASE, it's an enzyme. Enzymes starts with a P, proteins, right? Okay, and then centrioles, we just talked about them. They form the mitotic spindle. What are centrioles composed of? Microtubules, the 9 triplets of microtubules. Yeah? Are we doing so far so good? All right, so I think you'll do really well. Just a couple of things. As far as more content questions, we have the 2 chapters, 1 and 2. How many questions? 50. So the 25 questions from chapter 1 and 25 questions from chapter 2. Split pretty much down the middle. Okay. Chapter 1, planes and sections. What would those be? For example, give me the specific name of a section that yields externally equal right and left side. Mid-sagittal, beautiful. Mid-sagittal, right down the middle. Mid-sagittal is also called the median section. Mid-sagittal, externally equal, right and left, perfect, Damien. Now, what if you go off-center? Externally unequal, parasagittal. They're both types of sagittal sections. Sagittal would be the big title, mid-sagittal, median, right down the middle, parasagittal, off-side. Okay, stop me if you have any. We're good on that? Okay. Give me 3 names for a section that yields a superior and an inferior part. Cross, transverse, horizontal. Perfect. That yields a superior and an inferior. Okay, or I could say it yields a cranial and a caudal section. Remember, know those 2 parts. Okay. So then, let's see. What do you call a section that yields an anterior and a posterior part? Frontal or coronal? Corona, remember the little tiara? Frontal or coronal? Frontal equals coronal. I think a frontal gives you a front and a back, right? But we're going to say anterior and posterior. Anterior ventral, one more time, posterior dorsal. Anything at an angle is said to be oblique. Did I miss any? All right, regional terms, naked guy, I can't emphasize that enough. No, please, no naked guy. Now, levels of organization, kind of started with that after terminology. So here we have first the chemical level. I will ask you about the 4 categories of big macromolecules. Those would be what? Carbohydrates, lipids, and nucleic acids. Nucleic acids, DNA, and RNA, right? Okay. Then we have cells, which are measured in micrometers, different types of cells. Of course, that's chapter 2. Then we have tissues. What is a tissue? A bunch of similar cells, similar function. Let's talk about 4 types of tissue. Epithelial, covering light, connective, muscular, and nervous. The best way to learn a class like this for me is to make flashcards. Don't get the expensive ones unless you're independently wealthy, but they won't help you much anyway. go to Target, right? And hopefully they still have a sale. Not, you know what, don't even get cards, get a piece of paper, cut it up. On one side, you'll write down, frontal coronal, what does it mean, right? Make those cards. Will you ever use them? Probably no. But just the process of having made them, you're gonna read, trust me, I've taken a lot of classes in my years. Okay, so, and these two chapters really lend themselves to that. All right, so we're past cells, past tissues, then we have organs. Organs consist of all 4 types of tissues, usually. Then we have organ systems, 10 or 12, whatever they are. I want you to know them. I want you to know what they're composed of and a little bit what they do. Just the titles right underneath or above the organ systems. Case in point, the ones that might be a little bit tricky, integumentary system. Integumentum is a what? Skin and appendages, right? That's one of the next chapters we're going to study. How about which organ system functions to clear nitrogenous wastes? Urinary system, nitrogen. Yeah, respiratory would be, air and stuff like that. So those are the tough ones. Endocrine, hormone producing, you'll be fine on that. Okay, now, microscopy. A little bit of a problem for some people with that because it's kind of not as tangible. So microscopy. We have microscopic anatomy and we have gross anatomy. Microscopic anatomy, 2 types, light and electron microscope. Light microscopy, L for light, L for low resolution, low magnification, right? Okay. Electron microscopy, EM. Oh, bless you. We've got 2 categories. That would be transmission, TEM. What do you call the one that scans? I just gave it away. Scanning. So the scanning, SEM, will give you a beautiful 3-dimensional scan using electron beams, right? And transmission, we have cut sections, so you go right through. The electrons transmit through. What's an artifact? Distortion. In which electron microscopies might you encounter artifacts? All of them. Because we're preparing those tissues for viewing. So we have to fix them, gotta stain them, gotta cut them, whatever. Artifacts apply to all microscopy. Okay, I have a couple of questions from you about radiographic anatomy, or radiology. Just note that that would be a subdivision of anatomy where you're using imaging techniques. So we have the PET scans, MRIs, things like that. You don't, please listen carefully, don't need to know exactly what MRI is. No, it's a type of imaging technique. The catch-all term for that study would be radiographic anatomy or radiology. Keep it simple. Read it, it's kind of interesting. I know a lot of you are going into that. Okay, and then body cavities. Make sure you look at body cavities, right? So we have dorsal and ventral body cavities. Dorsal body cavities, we have for the brain, cranial, for the spinal cord, vertebral, right? So that's dorsal. Then we got the ventral anterior, right? Bigger, right? So then we have the diaphragm muscle, very nice division between thoracic cavity superior to it and abdominal pelvic cavity inferior to it, okay? Diaphragm. Now, the thoracic cavity. What is the mediastinum? The space between the 2 pleural cavities. 2 lungs, each within a pleural cavity. The space between the pleural cavities is the mediastinum. It's a space. What does the mediastinum contain? As the largest cavity, starts with a P, pericardial cavity. Mediastinum is a space between the lungs, space between the pleural cavities. It contains the trachea, contains the esophagus, a lot of the big vessels, but the largest cavity within the mediastinum, one more time, pericardial cavity. Okay, then we have the abdominal pelvic cavity, and more later on in the book, but we have the peritoneal cavities closer to the front, and the back organs are behind the peritoneal membranes, and they're described as being retroperitoneal. Abdominal pelvic cavity towards the front, peritoneal. Towards the back, without those membranes, would be the retroperitoneal space. Kidneys, retroperitoneal. Now, ventral body cavities, we just mentioned 3 that have names that begin with a P. For lungs, plural. For the heart, pericardial. For the anteriorly located organs would be peritoneal. Plural, pericardial, peritoneal. These cavities are defined by what? Serous membranes. Serous membranes. Each serous membrane has how many layers? 2. Forming the pared, the wall, parietal, sitting right on top of the organ, visceral. Parietal and the visceral, serous membranes. So we have parietal and visceral, plural membranes, parietal and visceral for the heart, pericardial, parietal and visceral for the anterior organs, peritoneal. 2 membranes, parietal and visceral, right? What's between those 2 layers? Serous fluid, right? Serous fluid. All right. I think I hit the highlights. If I missed something, but it's still in your notes, please look at them. Any questions on chapter 1 before we go to 2? Naked guy is the only thing I could think of that's a stumbling block. You know, naked guy. All right, so chapter 2. Chapter 2 pretty much just covered it, so it should be fresh in your mind. So that would be on cells. Let's start with the plasma membrane, also known as the cell membrane, also known as the plasmalemma. Cell, plasmalemma, plasma membrane, same thing, right? That's the external covering, okay? A lot of the organs also have a plasma membrane. Okay, so first of all, what are the structures within the plasma membrane that contain polar and non-polar ends? Phospholipids. Beautiful. Phospholipid bilayer. Each phospholipid has a polar, hydrophilic, and hydrophobic non-polar end. Okay? Phospholipid bilayer. And that always occurs. Then inserted into these phospholipid bilayers, we're going to have proteins. Remember, those are macromolecules. And 2 categories of proteins depending on how they are inserted or not into the membrane. The proteins going right in the middle or inside the would be integral, inside the phospholipid layer integral. The ones that are attached on either side are the peripheral, even on the cytoplasmic side. So not within the bilayer, but on either side. True or false, most integral proteins are transmembrane, go right through, absolutely. True or false, many of these proteins have channels. Yes, right? That's for stuff to get in and out of the cell. Okay. Cholesterol stabilizes. What do you call the externally placed sugar coat? Glycocalyx. The sugar coat and consists of glycoproteins and glycolipids. What does it do? Biological marker, cell recognition, maybe cell adhesion. Look at her, she is ready right now. Okay, good job, that's awesome. All right, so as far as movements across a membrane, all right, what is diffusion? Down the concentration gradient. From outside in only? No, either way. So when small molecules zoom right through that membrane from an area of high concentration to low concentration is called diffusion. In our case, it's really simple diffusion for the physiologists in the group. So diffusion, small molecules move down a gradient. What's osmosis, please? Water molecules, not big gulps, that would be pinocytosis, right? Water molecules zoom in and or out of the cell down their gradient, osmosis, easy peasy. Okay, then we have transport mechanisms. in transport mechanisms, there's different types. How many of you are taking physiology in the future? Yeah, so don't listen, or now, yeah, so don't listen too much to that. Okay, so when we talk about transport mechanisms, it's going to be much more detailed in physiology. So, but for our purposes, transport mechanisms are ways to get things into the cell or out of the cell using protein carriers or pumps, right? Carriers or pumps, that would be transport mechanisms. There's different types, facilitated diffusion and all that. Don't worry about that, right? Okay. Then we have the bulk or vesicular transport. Bulk, lots of stuff. A vesicle is a little bubble, right? And here we have either coming into the cell would be what? Endocytosis, leaving the cell. Exocytosis, reverse of endo, so that's easy. The endocytosis, 3 different types. Remember, we're making vesicles, right? First of all, cell-eating white blood cells, whatever is called phagocytosis. So we're making a phagosome, bringing in the E. coli or whatever, fusing it with a lysosome and dealing with that, right? So phagocytosis, cell eating. You're incorporating solid structures, particles into the cell, forming a vesicle. A phagosome is a vesicle. What's pino or pinocytosis? Cell drinking. You're still forming vesicles. A little bit of fluid with solutes in there. Now, as soon as you have a vesicle, you know it's endocytosis, because what's osmosis? Just water molecules, little teeny tiny ones, right? No vesicles involved. Receptor mediated endocytosis. Give me just one word. Specific. It's very specific. It's fishing. Yes, we're fishing for trout or whatever. Receptor mediated endocytosis is specific, and you're right too, because we have specific receptors fishing for a specific cargo of things that we're going to internalize. One more time. Receptor mediated endocytosis is the most specific. Why? We're using receptors to bring in a specific cargo of stuff. Beautiful. Okay. Anything on movements across the membrane? Either way. Oh, okay. Cytoplasm. Easy peasy. So cytoplasm, cytosol, the background fluidy stuff, inclusions temporary, components, and then finally permanent organelles, right? Pretty much covered all of that. Let's see, I think on the other one, what does the Golgi apparatus do? Packages proteins. Does it make proteins? No. It receives proteins from the RER, rough endophosphate reticulum. I think we covered all of the organelles. Anything in organelles? What is the cytoskeleton? Yeah, maintain shape, movement, whatever. So it's an internal network of miniature little tubes. Microtubules are those big hollow things. Not only do microtubules form the interior of centrioles, but they're also free-floating, not free-floating, but within the structure of the cytoplasm. Then we have microfilaments, would be actin, contraction, and intermediate filaments, intermediate in size and also for tensile strength. So that's the cytoskeleton. Okay, and then finally, the stuff that we just covered, let's recover it. So nucleus, nuclear envelope, 2 membranes, phospholipid layers. We have nuclear pores. We have then nucleoli, which do what? They make ribosomes, which then leave the little holes, the nuclear pores. Then finally, we have cell cycle. So when the cell is not dividing, that phase is called interphase. Inside the nucleus, chromatin. Okay, and then we have the 4 phases of mitosis, cytokinesis. Maybe this is easy. So prophase, if you remember that in prophase all of this stuff happens, right? Meta, you're going to remember chromosomes in the middle. Ana, apart. Telophase, reverse of prophase. And cytokinesis, kind of starts and anaphases, doesn't matter, we have that little furrow forming. Cytokinesis, we are divvying up the cytoplasm. It's not a perfect mechanism, okay? All right, any questions? Oh, we have about 10 minutes. Any questions on you guys? Yes?
Can you explain the mitotic spindle again?
The what?
The spindle?
The mitotic spindle? So the mitotic spindle, and the book will give you a lot more information. The mitotic spindle is like a bunch of little wires coming out. So in the mitotic spindle, I don't even know what I would draw for mitotic spindle. But basically, we have a bunch of microtubules that initially are within the centrioles. And you have that nice little triplet of, 9 triplets of microtubules. So then the microtubules and centrioles are just sitting there. When the cell is about to undergo cell division, and this starts in prophase, the centrioles will extend the microtubules. There's different parts, I know what you're getting at. There's different parts of the mitotic spindle, don't worry about it, okay? So once the microtubules extend from the centrioles, so all these gold, yellow things are microtubules, right? Check this out, the microtubules then ensure a very specific and orderly division of the chromatids into the opposing cells, okay? Typically, the microtubule, the mitotic spindle, different parts of it, don't worry about it, don't worry about the centrosome and all that kind of stuff, centromere, yes. But what, okay, let me see, so this is a microtubule. Where do you think it initially attaches to? The centromere. And then when the centromere splits, the microtubules will start to pull away the chromatids into the 2 newly forming daughter cells, children cells, okay? So mitotic spindle ensures a very uniform and orderly division of the 2 chromatids. By the way, once the 2 chromatids are formed, each one of these is known as a chromosome. Confusing. Chromosome could have either 2 chromatids. Once the centromere splits, you have the 2 chromatids. Each one is called a chromosome again. And then it's going to double up if the cell is going to divide and the process continues. What do you call that doubling of DNA stuff? DNA replication. Okay, good question. Okay, anything else? Yeah, make sure. Just a couple of things for folks arriving late. I don't want to scare you, but I don't want to surprise you at the end. If you come in 10 minutes, more than 10 minutes late, you cannot take the test, okay? And you might be able to take an essay format. Don't do that. Please, please be here on time for the exam. I know you're gonna rock it, and everybody's ready to go. Okay, all right. Well, I'll see you on Monday, everybody, and today, Wednesday, folks, tomorrow, Thursday. All right.